Hello everyone, my name is Kanil Kumar and today I'm going to represent an article which is based on cross-contamination between processing equipment and the delay meat by Listeria monocytogen. This is an overview of this article. So basically, Listeria monocytogenes is one of the pathogenic bacteria which frequently occurs in the processing of the foods or handling of the foods or while the transporting of the foods. So this kind of food can't be ingested which is having Listeria monocytosis because this kind of food, if we ingested, it can cause illness or sometimes it can cause serious illnesses too. This kind of bacteria are mostly found in the environments, soil, water, vegetation, and animals. The main thing which makes this bacteria very lethal is its surviving temperature. It can survive at very low temperature, like the refrigeration temperature, which is generally 4 degrees centigrade. So this is very pathogenic bacteria, very dangerous bacteria, which can survive at low temperature, and this kind of bacteria needs to be cured or eliminated during the processes. Symptoms may include a fever, muscle aches, nausea, vomiting, and kind of things. But if it goes severe, symptoms may include headache, stiff neck, confusion, and the loss of balance. So that's why this kind of bacteria needs to be tested and eliminated as soon as possible. This study was conducted to determine the three of the objectives. The first one is the dynamics of the cross-contamination of Listeria monocytogenes. This refers to the movement or the transfer of the Listeria monocytogenes from one equipment to another equipment or to the foods. The second objective is to study the influence of sample size taken for analyzing of the Listeria monocytogenes. And the third one is the fate of Listeria monocytogenes on sliced delay meats of different types of during refrigerated storage. So basically, an inoculum was placed at the equipment, and this inoculum was a mixture of five strains taken from the Frankfurters, coleslaw, Mexican style cheese, deli meat, and plantation. So at first it was 10 raised to 3, and other was also inoculated like 10 raised to 2 and 10 raised to 1 for other verification and other assays. So firstly they inoculated 10 raised to 3 CFU on the commercial slicer and did another kind of experiments which will be discussed in the following slides. Materials and method used for this research was like includes 10 microliters of the 5% host serum albumin containing 10 raised to 3 Listeria monocytogen was placed at the slicing blade. Furthermore, 10 raised to 1 and the 10 raised to 2 CFU were also placed to determine for the Turkey studies and also for the BAX PCR studies. Another samples were also observed, like the negative controls and the positive controls were also taken along with the test packages after the slicing. For the initial studies, meat samples were assayed at 1 and 30 days post slicing, whereas the later studies, after the post slicing, it was studied after the 60 days and 90 days of storage. The storage temperature was 4 degrees centigrade. Now, the detection of the Listeria monocytogenes in the meat samples. So they followed a series of chronological steps that is to be followed for the isolation and identification of the Listeria monocytogenes in the samples. So initially they took 25 gram of the sample from each package, and afterwards they tried to took the entire package. So there are two steps that went for the series of identification and isolation. One was from the 25 gram of sample taken, and another was the entire package. Both includes the same kind of procedure. The 25 gram of sample was firstly inoculated in the 225 ml of the vermouth broth and then followed by the zero point 0.1 ml of the enrichment culture before after incubating at a 30 degrees centigrade and 24 hours. Then after the isolation or the broth was further inoculated for the freezer with the freezer broth, and the loop of the enrichment culture was streaked on the Oxford agar. So likewise, there are series of dilution, series of streaking and inoculation were done to get last result, which was on the TSA-YA plate. The end result was the blue is hello under the Henry illumination color, which confirms the presence of the Listeria monocytogenes. And it can also form the catalase as it is capable to form a catalase enzyme. Afterwards, detection of the Listeria monocytogenes by the BIX in PCR assay was also done, as it needs to be compared with the enrichment culture. There are two types of steps. There are two types of procedure were followed to determine the detection of the Listeria monocytogenes in the food. The first one was the BX PCR, and the another one was the enrichment of the culture. Now, in the BX PCR assay, 100 microliter of the meat UVM enrichment culture was prepared and transferred to 10 ml of 3 uh, whatever the enrichment broth was of the MOPS LEV, and afterward it was included at 31 degrees centigrade for 24 hours. Afterwards, the another procedure were followed by the uh, bacterial cells, which were analyzed at 55 degrees centigrade for 60 minutes, and it was placed in the microtubes and the tablet containing the PCR reagent, which was then refrigerated at 4 degrees centigrade after the heating. Now these microtubes were sealed and tablet was dissolved, placed into the 96 well plate, assayed by the thermocycler and the fluorescent detector, and the results were recorded by the computer automatically. So basically the last step was done in the thermocycler and the fluorescent detector, which is usually connected with the computer, and all the analysis was done by the graph. And a graph the final graph can represent um, the dissolution power and the fluorescence change in the fluorescence color, which is detected by the computer automatically and represented by a graph. The next step would be a confirmatory testing of the Listeria monocytogenes positive samples. Colonies on the T side were confirmed by the BX PCR assay as mentioned above, like it is done by the two steps, which is the enrichment culture and the BX PCR assay. Then afterward, it is again strict into the MOX agar incubated at 37 degrees centigrade for the 24 hours. Now, then they assayed for the Listeria monocytogenes using the US FDA cis enrichment culture. So the last one colonies on the MOX will be the Listeria monocytogenes for the positive samples. Now the enumeration of the Listeria monocytogenes is done, which is obviously for the positive samples. 30 ml of the homogenate was liquid into the 3 tubes, twice serially diluted in 1:10 ratio and incubated at 37 degrees centigrade for the 24 hours. The direct plating can be done also for the positive controls as the sample of turkey meat held for the 60 and 90 days. A high number of growth is expected for the uncured turkey. And the next dilution was for the meat UVM homogenate was serially diluted again with 1:10 ratio in the Butterfield's phosphate buffer. The first time it was diluted with uh, the what can I say? It's a UVM broth, and now it is for the Butterfield's phosphate buffer. And then again it plated into the MOX plating, and the colonies were confirmed by the again BAX and the PCR assays. The final results were astonishing. Significant changes were observed in the turkey and salami, whereas very minimal changes were observed in the bologna. The reason behind these changes will be discussed further in the discussion board. But for as of now, let's discuss for the chemical changes. Salami contributed the lowest pH and the moisture amongst all after the refrigeration or the storage time. Whereas it contributed the lowest share in the fat percentage and the sodium chloride after the refrigeration. Turkey with a significant increase in microorganism. The reason behind this might be the higher percentage of the fat and the sodium chloride. The lower percentage of the fat and sodium chloride and the higher percentage of the moisture might be the reason behind the increasing of the Listeria monocytogenes. However, the proper reason behind this will be discussed in the discussion. Now, the detection of the monocytogenes on the DELI meats. The greatest number of the Listeria monocytogenes were obtained in the oven roasted turkey after the storage at 4 degrees centigrade, whereas the salami and the bologna sample was decreased after the refrigeration storage. Improved detection was observed by increasing the size. So basically, two sizes were taken, 25 gram and the entire pack. So in the 25 gram, the detection was quite very lower or negligible. But when took the entire slice, we got the proper reading and the proper detection. The reason behind this would be the contact of the delimits and the slicer. If the contact of the delimit is in another part, then if we are taking the sample, the other part in which the contact has not been made, then it won't be able to get we won't be able to get the result or the detection of the Listeria monocytogenes. So it is suggested that the entire package should be taken for the evaluation. The larger sample size did not yield an increased number of the Listeria monocytogenes positive samples for the salami and bologna, because there might be some preservatives added which can act as a growth inhibitor for the Listeria monocytogenes, and the further pH change in can also contribute towards the decreased growth of the Listeria monocytogenes. Now, the efficacy of detecting L monocytogenes by the BX PCR assays and the enrichment culture. From the table, we can see that at the initial days, like from day 1 to 30, for both of the inoculum, for the 10^1 and the 10^2, there were no growth of the Listeria monocytogenes, or you can say the no growth for the positive samples. But after 30 days, the storage time at 60 days and 90 days, we saw a minimal amount of growth of Listeria monocytogenes. So for the 10^1 colony forming unit, 3 and 5 was observed after 60 and 90 days, whereas in the 10^2 colony forming units, we observed 7 and 33 CFU at 90 and 60 and 90 days respectively. Now the detection of the Listeria monocytogenes on DLI plates will be remains the same. Colonies of the T cells plates will be counted and again will be streaked on the MOX, and it will be again detected by the positive BX and PCR assay. However, for the two samples in which Listeria monocytogenes was detected only by the BX assay, the pathogen was not detected in the corresponding MOPS lab brought by the USDA enrichment culture procedure, which was firstly inoculated by the UVM. Now, the positive samples in salami and the bologna decreased during the storage because not detectable as a positive control 60 days or 90 days, because might be the reason of the chemical change in the chemical properties. The last but most important part, the discussion. Adherence and survival of the Listeria monocytogenes was well studied, because the adherence can be described as the location of contact of the slicer, slicing equipment, and the meat could play a significant role in determining and the detection of the Listeria monocytogenes. However, the dynamics of the cross contamination have not been reported. This represents that if the sample is inoculated in one part of the blade, and the delay meat or the any kind of meat has been contacted with the slicer, how this inoculum which has been inoculated in the which has been spread on the blade can transfer from slices to meat and other equipments also, like it can be transferred from slices to conveyor belts to side of the conveyor belts, and similarly we can get a series of samples which shows the positive samples for the Listeria monocytogenes. So this was not thoroughly studied, and that's why the dynamics of the cross contamination have not been properly reported. But the study revealed it could be the transfer from the commercial slicer, degree of transfer, the number of Listeria inoculated into the slicer was properly studied. Like for the 10^1 and 2, 10^1 and the 10^2 colony forming unit, when inoculated, no significant number were observed. But however, when the higher amount of the inoculum were inhibited, like 10^3, it shows some detection. So greater the number of positive samples, larger the sample size of the turkey meat. Fewer the Listeria monocytogenes positive samples were obtained from the salami, because the reason behind this is the lower moisture content and the pH value. Lower moisture doesn't allow the Listeria monocytogenes to grow, and it also does not grow at the acidic condition or the low pH value. Now for the bologna, it contains the lactate and the diacetate, which can act as a preservative and inhibit the growth of the Listeria monocytogenes. Cell numbers increase on the bologna. However, the cell number were increased in the bologna with the preservative on the elevated temperature. So here we can say temperature can play a very pivotal role in determining the shelf life also and the number of cell numbers of the Listeria monocytes after the refrigeration period. So we can conclude that the sample size for the detection or the evaluation, storage temperature, and the amount of inoculum initially present plays a very important role in deciding the number of cells or the CFU in the sliced D limates. The last but not the least, the Beck's PCR assay appeared to be an acceptable alternative to the US FDA enrichment procedure for detecting L monocytogenes in the pre-cooked delight. Thank you for watching this article. I hope you got, and any questions related to these articles are welcome. Thank you.